Assalamualaikum and good morning to Madam Hatia. My name is Nur Farafika binti Razwan. My student ID number is 42010011421. The topic that I've got for my video presentation is stool culture and sensitivity. I would love to show you how the sample being processed in Hospital Raja Permaisuri Bainun Ipoh. What is stool? Another name for stool is feces, made from what is left after our digestive system absorbs nutrients and fluids from what we eat and drink. Our stool passes out from our body through our rectum and anus. This is Bristol's stool chart. There are 7 types of stool. Did you know that color of the stool can give us hint of abnormal bowel movement? Sample will be collected in specimen container and carry Blair medium transport. Specimen container must be dry and clean. Carry Blair medium transport is usually used for pediatrics patient. Sample must be labeled correctly with patient's details and tally with their request form. Sample collection. First, you need a sample container. Then, you must label the container with the patient's details and test requested. Next, take the sample and put inside the bag sample. And last, make sure you wash your hands. Tests will be done in microbiology lab to detect and identify the bacteria that causing pain to patients so they can get treatment. Sample will be taken before antibiotic treatment were given. Common pathogen for stool sample is Escherichia coli, Salmonella species, Vibrio cholera, Campylobacter jejuni, Aeromonas species, and Escherichia coli O157H7. McConkey agar is a selective and differential medium. It can isolate bacteria based on their ability to ferment lactose. XLD agar is a selective differential medium. It is to isolate gram-negative pathogen from stool specimens and others, more suitable to isolate Salmonella and Shigella species. Meanwhile, Aeromonas agar is a selective medium and it is to identify Aeromonas species in food, water and other sanitary importance. TCBS agar is a selective medium to isolate Vibrio cholera and other Vibrio species like Vibrio parahemolyticus. Campylobacter agar is a selective medium for isolation of Campylobacter jejuni from stool samples. It is an enriched selective blood agar. Sorbitol McConkey agar is to differentiate Escherichia coli and Escherichia coli type O157H7. This is the result of the pathogen after culture in each agar. Selenite F, a selective enrichment for the cultivation of Salmonella species, can be enrichment medium for isolation of some Shigella species, recommended for the transport of strains of Vibrio species because they can survive 2 to 5 days in sodium selenite broth. Alkaline peptone water, enrichment and transport media for enrichment of Vibrio cholera and Vibrio species from water, food and other clinical samples, can be used for direct microscopic examination like hanging drop method. Identification test. Firstly, we will do the triple sugar ion biochemical set test, and then oxidase test will be done on certain suspected organisms like Aeromonas species and Vibrio species. This picture shows the identification charts for stool pathogen. Confirmation test. Sampel sent to Makmal Kesihatan Awam Jelapang Ipoh. Only for sample who suspected with Escherichia coli species and Salmonella species. Serotyping test for Escherichia coli is to differentiate E. coli strain EPEC and EHEC. It is complex, and there are two main groups of serotypes. E. coli is classified into 150 to 200 serotypes based on somatic, capsular, fimbrial, and flagellar antigens. Serotyping test for Salmonella. The outermost portion of the bacterial surface covered with lipopolysaccharide called the O antigen. A slender thread-like structure called the H antigen is a part of flagella. Each O and H antigen has unique code number. Salmonella has many types of serotypes. Each serotypes are found all over the world, can cause severe illness when infect people, while others can cause milder illness. These are the examples of molecular typing techniques that has been used in some laboratories in whole world. Sensitivity test is a test to determine sensitivity of bacteria to an antibiotic. It determines the ability of drug to kill the bacteria. Doctors can determine the right antibiotic treatment for an infection and to monitor changes in bacterial resistance. This is the list of antibiotics that being used to treat patients that infected with Salmonella species and Shigella species. This video shows how you should do the sensitivity test. After you open the cap and dip cotton swab inside it, wipe swab on plate gently. Rotate plate and continue swabbing to make sure it covered the whole plate. Now, mark 4 quadrants on plate and transfer this to the plate.